Hello class. So today we're going to cover DNA replication. This is lecture 6. So let's start off with a little background. DNA replication is essential for all organisms, right? We all have to copy our genetic material. And DNA replication is the duplication of a genome's organism. The process of DNA replication occurs very rapidly. The polymerase itself can actually travel down DNA quite fast. And you can sort of stop and think about that for a second. We have 3.2 times 10 to the ninth billion, or 3.2 times 10 to the ninth nucleotides of DNA that needs to be copied, so it has to move very fast. Also, DNA replication must have a very low error rate, right? You don't want a situation where every time our DNA is being copied, we're introducing a ton of mutations. That would just be very bad. The mutation rate during replication for mammals is 1 times 10 to the ninth, and we'll actually look at where this number comes from. It's actually a combination of things. And replication is actually a pretty highly conserved process from bacteria all the way up to humans, and so studies in bacteria is pretty relevant to humans. And just a few things that I think are sort of important. I'm sure you guys have all replicated DNA, or some of you have replicated DNA in the laboratory using the enzyme Taq polymerase. So if you've ever set up a PCR reaction with Taq polymerase or another polymerase, you've basically duplicated DNA. And the whole reaction occurs through a tail condensation reaction, and we'll look at that. And that's actually quite important to the low error rate that we see, right? If you didn't have tail condensation, the error rate would by default have to be much higher, and we'll see why that is in a few slides. So just a little continuation with some background. We've talked about this before. But what you have basically in a mother versus a daughter is when two gametes come together, they form a zygote. Okay, and that zygote can then give rise to somatic cells, or you can actually have it produce more germ cells, which then the germ cells can then come and form a whole nother offspring. Okay, where you get the zygote and the somatic cells. So germ cells basically transfer genetic material to offsprings versus somatic cells, which basically are all the other tissue in your body. Germ cells have 1N genomes versus somatic cells, which have 2N. Okay, that means that there's basically 2 copies of each chromosome versus 1 copy of each chromosome in the gametes. Okay, so I'm sure you guys all know this too. DNA is copied from a template strand. So what ends up happening is you have your double-stranded DNA, and it's actually separated, and then the template strand is used to synthesize the new strand. Okay, and so both of these strands right here, the S strand and the S prime strand, become template strands during replication. And the enzyme that actually adds all of the nucleotides during replication is called DNA polymerase. The chemistry of base addition is actually quite simple, okay, but there's certain things that you guys really need to know here. And one of it is that synthesis occurs from 5' to 3', being read off the template strand from the 3' to 5' direction. And this is actually really important for the proofreading activity, as we'll see. But what you have is complementary base pairing between an incoming deoxyribonucleoside triphosphate, which is right here. So here you have G base pairing with C and the hydrogen bonds forming. And then on the sort of growing DNA primer strand, you get basically addition on the 3' carbon with the phosphate. Okay, of the incoming nucleoside triphosphate. And this actually forms what's called a phosphodiester bond. And in the process, it releases pyrophosphate. These are two phosphates connected together, basically. And so you get this pyrophosphate released, which then is broken down into two inorganic phosphate molecules. Here's just another little schematic of how the whole process, how DNA polymerase works. You have your sort of your primer strand, and it's using tail condensation to add nucleotides in the 5' to 3' direction. You have your incoming nucleoside triphosphate coming in, and that actually releases pyrophosphate, which is then broken down into two inorganic phosphates, and it grows in the 5' to 3' direction, being read off the template strand in the 3' to 5' direction. Okay, so here's just a little sort of schematic of it where you basically have, you know, the enzyme you can sort of think of as a hand here, and the incoming nucleoside triphosphate comes in. There's a repositioning event and a little conformational change, and then the phosphodiester bond is formed and pyrophosphate is released. And so ultimately what's going on here is that bases are being added to the DNA one at a time, okay? And the energy of the base addition actually comes from the release of the pyrophosphate. And so ultimately what you have is this process called semi-conservative replication, where you have basically just a single copy of DNA. It's separated, and then the new strand is actually then associated with the old strand, and you get two, and then once again those strands can be broken, and the new strand always goes with the old strand. Okay, and that's pretty much it. And this just goes to show you sort of the logarithmic replication that occurs when you do a PCR reaction. Now, one of the things with the semi-conservative replication is that it forms these replication forks. Okay, here's a nice little schematic of the replication forks, and here's just a circular piece of DNA, and you can see the replication forks here. So this is an electron micrograph of DNA being replicated, and you know, here is basically a schematic image of it. Now, because the replication occurs in the 5' to 3' direction, the replication forks here create a leading and a lagging strand. And so I just told you that replication can only occur in the 5' to 3' direction. So then the question is, how is the second strand copied? And if you think about the directionality of DNA, this isn't necessarily a trivial issue. Okay, and the way it's handled is you have what's called a lagging strand. Okay, so here's your leading strand, and once the leading strand starts, and the polymerase is traveling from 3' to 5' on the template strand, it can synthesize in the 5' to 3' direction, and that can just continue on, right? There's no problems there. However, on the lagging strand, you actually synthesize these fragments called Okazaki fragments, okay? And that, once again, because you have to go from 5' to 3', these have to go out. It can't just go along. So the polymerase is actually feeding the lagging strand out, and then these Okazaki fragments are actually being synthesized, and then they're ligated together. Okay, and this whole process is called backstitching. And the Okazaki fragments, depending on the organism, can range in size. In eukaryotes, they're usually about 1,000 to 100 to 200 base pairs, and then in bacteria they can be 1,000 to 2,000. Now, one of the really neat things about DNA polymerase is that it has proofreading ability. Okay, it can tell most of the time when it puts a bad base in. Okay, and DNA mismatches are pretty common. And they actually need to be fixed to sort of maintain this 1 × 10⁹ error rate, right? This really, really low error rate. And so one of the things that can happen is you can get a tautomeric form of C, which can base pair with A, and we'll look at the chemistry behind that in just a second. But what if you want to just sort of follow the schematic over here? You can have all different forms of, nucleoside triphosphates over here, and if you have your primer strand copying a stretch of A's, for example, occasionally a tautomeric form of cytosine can come in and base pair with an adenine. Okay, this tautomeric form isn't going to last for very long, and then eventually you won't get the good hydrogen bonding, and the base will not be base paired. And this can be sensed by the DNA polymerase. And then the DNA polymerase actually has 3' to 5' exonuclease activity that can remove the improperly paired base, and then the correct base can be put in. And so let's just look at the chemistry of this. So here's normally your cytosine. It can tautomerize. So one of the bonds actually goes up to, so you've got this double bond at the nitrogen, and then you have a hydrogen bond here, and then you can form this hydrogen bonding with an A base. But this doesn't last long. It's a somewhat of a rare event, but it can occur. One other problem that can occur is G can form 2 hydrogen bonds with T really easily. So occasionally a G will get put in place of an A during DNA replication. So anytime there's these sort of mismatches that get put in, the exonuclease activity of the DNA polymerase itself can chew back the end, and the proper base can be put in. And here's just a little schematic of how the exonuclease proofreading of the polymerase works. Okay, so here you have the enzyme, the DNA polymerase adding a nucleotide, and if it's put in the wrong base, it actually flips out to sort of an editing site where then the exonuclease activity removes the wrong base and the other base can be put in. Okay, and once again, the exonuclease activity occurs in just the 3' to 5' direction. And so here's a nice little movie that sort of shows that, and hopefully I'll be able to play the movie here. I'm having a little bit of issues playing the movie while recording. You guys sort of ran into this last time, I think. So let me escape out here. Oh, there we go.
DNA polymerase faithfully replicates DNA by using the nucleotide sequence of the template strand, colored gray, to select each new nucleotide to be added to the 3' end of a growing strand, colored yellow. The different domains of DNA polymerase are colored differently. Before a nucleotide can be incorporated into DNA at the 3' end of the growing strand, the blue finger domain of the polymerase moves inward to correctly position the nucleoside triphosphate. A pyrophosphate group is released when each nucleotide is added. In this view, the details of nucleotide selection at the active site are shown, with the incoming nucleoside triphosphate and the template nucleotide in light blue. The growing strand is green, and the template strand is red. When the finger domain moves inward, the nucleoside triphosphate is tested for its ability to form a proper base pair with the template nucleotide. When a base pair forms, the active site residues catalyze the covalent addition of the new nucleotide onto the 3' hydroxyl group on the growing strand, and the entire process repeats at speeds up to 500 nucleotides per second. On rare occasions, approximately once every 10,000 nucleotide additions, the polymerase makes an error and incorporates a nucleotide that does not form a proper base pair onto the end of the growing strand. When this occurs, the polymerase changes conformation and transfers the end of the growing strand to a second active site on the polymerase, where the erroneous added nucleotide is removed. The polymerase then flips back to its original conformation, allowing polymerization to continue. As a result, such a self-correcting DNA polymerase will make a mistake only about once every 10 to the 7th to 10 to the 8th nucleotide pairs.
Okay, so you guys can actually watch that video. There's a lot of great videos on YouTube, and you can find them. That was actually, I believe, from the Howard Hughes Medical Institute. So if you just Google those, you can watch them again, or you can watch them here. Um, okay, so ultimately, when you have polymerization, and by the DNA polymerase, you get an error rate of about 1 times 10 to the fifth. Okay, and so it's not perfect. And by itself, if our DNA polymerase was only this good, we'd actually suffer a lot more diseases. Um, because this just isn't sufficient to copy the amount of DNA that we have. Okay, but when you incorporate the exonuclease activity in the proofreading, that goes up by quite a bit. Okay, it goes up to basically 1 times 10 to the seventh. But that doesn't get us to the 1 times 10 to the ninth that we need. And in the second part of today's lecture, I'll talk about the strand-directed mismatch repair, which really brings the enzyme or brings the replication process itself to 10 to the ninth, even though the enzyme is only about 1 times 10 to the seventh, has the accuracy of 1 times 10 to the seventh. Okay, so we'll look at this later on. So there's just a bunch of different methods by which the cell uses to actually do a really faithful job of replicating DNA. Okay, so now we want to get to the point where we really understand why DNA is replicated from the 5' to 3' direction. Okay, and part of it has to do with the proofreading activity. Okay, so if DNA polymerase went in the 3' to 5' direction, or if synthesis went in the 3' to 5' direction, there'd be no energy for proofreading. Okay, after the exonuclease was removed, and that's why DNA polymerization undergoes tail condensation. All right, and we'll look at this. Okay, so let's say we're going in the 3' to 5' direction. All right, the energy for the base addition would have to come from the existing strand. All right, the release of this pyrophosphate group. Okay, so when an incoming nucleoside triphosphate got added, the pyrophosphate here would be released and the new base would be added. Now, if it turns out that this is the incorrect base, it would then be removed by the exonuclease, and then there wouldn't be any more energy to add the next nucleoside triphosphate, and it would just basically stop. So if our polymerase went from 3' to 5', or if synthesis was happening 3' to 5', you basically couldn't have any proofreading activity in the enzyme. And if you look in the other direction, if we go 5' to 3' with the growing strand, the nucleoside triphosphate that gets added, the energy is actually coming from it. Okay, the pyrophosphate's removed from the nucleoside, the incoming nucleoside triphosphate. And so then when it gets removed, if there's a mismatch right here, you can just bring in a whole new one and the energy's coming in. So once again, you just need the proofreading activity to go from the 5' to 3'. You need to be going in the 5' to 3' direction for the proofreading activity. And I hope this makes sense. And once again, this is understanding the directionality of DNA and why DNA is copied in this manner is really important for you guys to know. And so make sure you understand it. Okay. All right, so now we're going to look at some of the accessory enzymes that are involved in DNA replication. Okay, one of the enzymes is this enzyme called DNA primase. Okay, you actually need a primer to start the whole polymerization process. And I know some of you guys have probably set up PCR reactions and you know you have to add your primers in. Okay, but in the cell we have this enzyme called DNA primase, and it actually makes an RNA primer of about 10 nucleotides long. And this primer is used to not only to start the leading strand, but it also is used to start the lagging strands or the Okazaki fragments. Okay, and so DNA primase comes in and it just creates this RNA primer in the 5' to 3' direction. When the lagging strand is actually synthesized, because it's synthesized in small fragments, these Okazaki fragments need this enzyme called DNA ligase to sew them together, right? So the Okazaki fragments started from the RNA primer, and then as the polymerase is moving down, it actually chews up that RNA primer to get rid of it. Okay, and then once you have the sort of the DNA polymerase finishing out by removing the old RNA, you're left with this nick that's right here. Okay, and the nick is sealed by this enzyme that joins the Okazaki fragments together. Now, a question that you guys might wonder is, why are they using an RNA primer and not a DNA primer? And the answer is that basically the creation of the primer is more error prone, and so it's better to basically build an RNA primer that's removed than to have a DNA primer where potential errors are put on by the primase. Okay, it's once again helps to lower the error rate of DNA replication. Here's how DNA ligase works, and if anybody has set up a ligation reaction in a lab, you probably have used this enzyme. In my lab, we use it quite a bit. You can buy it from a whole bunch of stores. But the way it works is you have your 5' phosphate and your 3' OH group, and DNA ligase uses ATP, and an AMP is added to the 5' phosphate to create this DNA ADP complex. Okay, and then the phosphodiester bond is formed by DNA ligase, and the AMP is actually released, and the nick is sealed. And so ultimately, DNA ligase uses ATP to seal the nick. And if any of you have ever used DNA ligase and haven't had much success with it, it's oftentimes that the ligase buffer, which has ATP in it, the ATP has gone bad. So sometimes if you just spike in some ATP into your ligase, it helps your reactions quite a bit. Another enzyme that's really important for DNA replication is this enzyme called DNA helicase. And you can sort of think of DNA helicase as the thing that's unwinding a zipper, okay? And there's a lot of energy stored in the hydrogen bonds that occur between DNA. And for example, if you want to break those hydrogen bonds, the amount of thermal energy that it's going to take is you're basically going to have to heat the DNA to 100 degrees Celsius. But DNA helicase actually uses ATP to sort of unzip the two strands of DNA. And this enzyme can actually travel very fast, 1,000 nucleotides per second, as it's sort of going down the DNA. Here's the structure of DNA helicase. Here's just sort of a schematic of it. Here's the ribbon diagram of it, and here's a nice little space-filling model of it, where it's sort of traveling in the 5' to 3' direction down the DNA. And it has 6 subunits, and it uses ATP to help separate. So the ATP hydrolysis is where the energy comes from. Now, when DNA is being copied, especially on the lagging strand, one of the things that can happen is you can get these DNA hairpins, okay? And these DNA hairpins can cause a big problem for for DNA polymerase. And so what prevents them on the lagging strand are these proteins called the single-stranded binding proteins. And they help to basically straighten out the DNA so that the polymerase can travel unabated down as it's copying it. And these hairpins actually will require a lot of energy to break down. Here's the structure of the single-stranded binding proteins. Here's just sort of like a schematic of it with the DNA sort of sticking out. There's 2 domains. And here, if you're sort of looking down it with the ribbon diagram, it just holds the DNA in single-stranded form. And these become very important as the lagging strand is actually looped out of the whole replication complex. Another important enzyme in all of this is the sliding clamp. Okay, and in general, the DNA replication requires a lot of subunits working together to basically allow the replication to proceed in a really rapid manner. And what the sliding clamp does is it keeps the DNA polymerase from falling off the DNA as it's copying the DNA. And the rate at which polymerase can fall off without the sliding clamp on the lagging strand is actually pretty high. And when you're synthesizing the lagging strand, when DNA polymerase comes into contact with the RNA primer and starts to get rid of it, this sliding clamp can then fall off. Okay, so when it hits the next piece of DNA, the sliding clamp just falls off. Now, the sliding clamp is actually loaded by this enzyme called the clamp loader, and the clamp loader is needed to add the sliding clamp onto the initial leading strand, and then every time a new Okazaki fragment is started, the clamp loader loads the sliding clamp, and then the polymerase can come in and you can synthesize the lagging strand. Okay, so this happens every time an Okazaki fragment is being created. Here's how the loading works. You basically have your sliding clamp and the clamp loader. The clamp loader uses ATP to load it onto the DNA, and then the ATP is hydrolyzed. The clamp loader falls off and DNA polymerase comes in, and then you can start synthesizing the DNA. So the clamp loader uses ATP to load the sliding clamp, and then DNA polymerase can come in. When you put it all together, this is what you get, and this is one of those things I really want you guys, and I've said this more than once already this term, it's the big cellular processes that I want you to know. So I want you to be able to explain from start to finish how DNA replication occurs, right? And so what you can see here is you have the DNA helicase that's separating the DNA double helix from the parental copy. On your leading strand, you have the sliding clamp sort of pushing along the DNA polymerase, and that's just sort of adding nucleotides unabatedly. On the lagging strand, you're synthesizing Okazaki fragments. Okay, so the DNA primase comes in, it adds an RNA primer. Once the primer's there, the clamp loader will load the sliding clamp, and then DNA polymerase is added on, and you start to synthesize the new Okazaki fragments. Once you get to the end of the Okazaki fragments, the RNA primer is removed, and then this nick is sealed by DNA ligase. And as the helicase is sort of looping out the lagging strand, it's bound by single-stranded binding proteins that help prevent it from forming those DNA hairpins. Okay, so once again, just I really want you guys to be able to explain how this whole process works, and I also want you to understand why DNA is synthesized in the 5' to 3' direction. Okay, and if you know that DNA is synthesized in the 5' and 3' direction, you'll know basically how the DNA polymerase is reading, right? It's reading in the 3' to 5', okay, because it's synthesizing in the 5' to 3'. If you think about it, it will all make sense and you guys won't be confused. And it needs to go in that direction because it needs the energy from the incoming nucleoside triphosphate and the release of the pyrophosphate to have that sort of proofreading, because if it ever has to remove a mismatch, it can use its exonuclease activity, remove it, and put another one on. Okay, so just know those types of things. Those are really important. And so if we take a look at this, this is a really great schematic, because if you look at an electron micrograph of the bacteriophage T4 replication machinery, you can see it's basically this, right? It looks, and you can sort of see where you have the leading strand, the newly synthesized strand, and then you have the strand that's looped out with where the Okazaki fragments are being made, and then your new sort of lagging strand. And so if you guys want to just see a movie on how all of this works, we can just sort of watch it. And once again, you'll be able to download these videos and watch it. And after this video, I think that's the end of part A of this lecture. So,
during DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
okay, so that was a pretty good movie that really sort of explained everything, that you just need to know about DNA replication. And so in part B, we're going to look at how the actual replication process starts and also some mismatch repair. And so I'll get started on that in a second. Okay?